All right, let's finish up chapter 8 here with enzymes and metabolic pathways. So, enzymes are proteins which speed up the rate of chemical reactions. Most enzymes are proteins, not all, not all. Some are made up of RNA. We are also going to talk about the energy of activation. How can we get these chemical reactions to start? So, enzymes speed up chemical reactions and they bind to specific substrates. The binding of a substrate with an enzyme causes a change in the enzyme shape and reduces the activation energy of the reaction. So here's our enzyme. We can see the binding pocket, which is right here where the substrate would bind. In this case, it bound. This enzyme then broke it in half. Generally, when they bind, they change shape. In this case, they would change shape to put stress on those bonds and to break it. So how do enzymes work? So when we talk about an enzyme, we can see here a lot of people are lactose intolerant. We tend to call enzymes after the substrate they attach to and add an ASE. So the enzyme we'll be talking about is lactase. You can see that right here, lactase. So lactase helps to speed up the rate of this chemical reaction so that you can correctly digest dairy products. Without it, you break it down too slow and it causes a lot of digestive problems. So lactose is a disaccharide. Look at the big hill we got to get up. Okay, that is called the activation energy. All right. So we've got to get up and over this hill in order to release that energy as we break that bond. Notice it is an exergonic because we're starting higher than we're ending. But we got to get up over that hill to get this started. And that hill is called activation energy. Now down here, you can see at the bottom, we have lactase. It binds to that binding pocket, lactose. It changes its shape and it breaks those disaccharides down into two monosaccharides. And it is also, you can see it's still an exergonic reaction, but that hill is so much lower, it doesn't require as much energy to get up and over. And it makes those chemical reactions go faster. So we can see here a graph indicating the red line is the amount of energy you're going to have to put in without an enzyme. And then the black line is with an enzyme. And you can see that activation energy hill is so much smaller. So this reaction will proceed forward faster. So again, here's the active site. We can see this little model here where that substrate would bind. Almost all all enzymes again are proteins. We can see just that picture there. So things that affect the rate of enzymes are, well, we really need to have those collisions occur, right? We need that substrate to run into the enzyme and not just run into it, but to actually run into that and bind at the binding site. And so if you have more substrate, you're going to increase your number of collisions and that chemical reaction will go faster. Temperature. All right. Temperature will also speed up, right? The molecules we're looking at will have more kinetic energy as they're moving faster. But the problem is if we move not so much moving too fast, but enzymes work at a specific temperature and pH where they work the best is called the native environment. Enzymes are designed to work the best in the environment in which you would find them. So, for example, our enzymes would work best at body temperature. A potato, so potato catalase works better at a slightly colder temperature because we find potatoes in the ground where it's cooler. We've got our thermoacidophiles that would work best in a hot acidic environment. We have some enzymes that work best in an acidic environment like our stomach or inside our lysosomes. So the closer you can get to those native environments, the better and it's more of an optimal rate for those enzymes. Again, if we have more enzymes as well, we are going to increase the frequency. Okay. We don't want too many enzymes in there just running into each other. All right. So it's kind of a sweet spot there. Cofactors, some enzymes require cofactors or what we also call coenzymes, both of those, in order to work properly. Cofactors are inorganic elements, like basically they are your minerals. So whether you're going to find those on the periodic table, they're going to be elements like iron, zinc. All right, those things are specifically those two are required for optimizing certain enzymes. Coenzymes are your vitamins. They're going to be an organic molecule like vitamin C, D, E, K. All right, so all of those things are going to also be needed for some enzymes to work at their highest rate. When we add a phosphate to another molecule, it's called phosphorylation. We phosphorylated it. Enzymes called kinases specifically are going to transfer those phosphates to other proteins, and often it activates them. It's like kind of like an on switch. So you can see here the substrate. We're going to get the phosphate from ATP. We're going to go to a kinase. The kinase is then going to phosphorylate the substrate and turn that substrate on. All right. Sometimes it's enzymes we turn on. There's all kinds of different things that we can turn on. Generally, they're almost always proteins. To regulate enzymes, there's two types of inhibition we're going to talk about. One is competitive inhibition, and this is where we have another molecule that has a very similar shape to the substrate. And so this here's what's supposed to happen. This little uh, funky shape is supposed to fit in there, and then it breaks it into two different pieces. Notice that this shape closely it's pretty much the same thing. So it fits in and it's going to sit in there for, how much time. And it's going to block the substrate that's supposed to be in there to make that product. For the most part, these things will pop in and pop out and then pop in and pop out. There are a few things that once they're there, they're not going to let go. One of such things is carbon monoxide. Carbon monoxide resembles carbon dioxide, and so carbon monoxide will bind to the hemoglobin in your red blood cell, and it does not let go. And so once you get so saturated with carbon monoxide, you're not going to be able to transport oxygen throughout your body. And so that would be kind of like game over. Non-competitive is where we have a secondary site on that enzyme and a different type of molecule is going to bind to it. When that molecule binds to it, so if we see down here, it binds to it, notice the shape change. And since the shape of the binding pocket is no longer the same, that cannot either fit or it won't let go. All right. So that is a way that we can kind of turn that enzyme off. So competitive will bind to the same site as the substrate. Non-competitive binds to a secondary site on the molecule. Both of which, you can see here, here's our competitive, here's our non-competitive, both of which is going to slow down the rate of that enzyme's productivity. We also have something called allosteric control. And it's kind of similar to what we just said about the non-competitive, right? Allosteric control is when an enzyme changes shape. The shape can either be turned on or off. So for example, in this particular, there's our quaternary structure, right? We have multiple proteins fitting together to make one functional protein. And so when we have this molecule here, okay, it is going to stabilize the active form. When it's not, and we have this other type of protein, it changes its shape into an inactive form. So we have this enzyme on or off. So we can see here when one binding site is occupied, the other sites are also change to receptive state and their affinity. Affinity means how much they like for that substrate increases. So once we bind there, we have those other binding pockets. Notice how it's changed shape. And now we have those other pockets available for more substrate. We also have something called feedback inhibition, and this is where enzyme inhibitors are important in regulating cell metabolism. In some reactions, the product may act as an inhibitor of one of the enzymes in the pathway, and this is called feedback inhibition. So if we look here, we have these yellow spheres, and its job is to bind to the enzyme. The enzyme then changes it to the orange pie. Now, notice we have the secondary binding pocket. So once we have enough orange pies, basically what we're doing is the concentration increases, so it's more likely that it will bind to the secondary site, and it tells the enzyme, nope, we're good. We have enough orange pies. We don't need any more. All right. And then, of course, as these move off and are used for the next chemical reaction, then your concentration goes down because, again, these don't stay in. And now that binding pocket will open up and we'll be able to make more orange pies. Okay. The location of enzymes. It is important to note that one enzyme does only one task. And many metabolic processes need multiple steps to be done. So, in order to maximize efficiency, enzymes are organized inside the cell to be close to one another. They're either contained inside the same organelle or they could be located next to each other in the cytoplasm by using the cytoskeletal support system. And then here's another neat little movie on entropy. All right, if time, which you have time on your own, it would be really cool if you watch that particular video on the universe and entropy. All right, that is it for chapter 8. If you have any questions about the content, again, just write them down and bring them to class and we'll discuss. All right. And that is it for chapter 8. I will see you in the next set of videos.